Hello, everyone, and welcome to NHF's annual Bleeding Disorders Conference. This session will start shortly. As we wait, we would like to thank the generosity of our 2021 sponsors. Without their support, this event would not be possible. Our platinum, gold, and silver sponsors are listed here. So welcome to the ATHN Advancing Care for the Blood Disorders Community session. My name is Kate Naumacher. I am the Vice President of Education at the National Hemophilia Foundation. I'm really excited to have this amazing session from ATHN, from our colleagues there, to really share some updates from this team. With that said, a few quick items for navigating this session together. There is no chat or Q&A for this particular session today. So if you have questions, we totally understand, but you can contact the speakers directly via email, and their emails will be listed at the end of the presentation. In addition, this presentation will be recorded and will be available to watch online on the conference platform after this session. Finally, please make sure to fill out the evaluation and give feedback for how we can improve this type of session and more sessions for next year's Bleeding Disorders Conference. We really do look at that information. And with that, I am going to hand it over to the ATHN team. Thank you all so much for being here.
Welcome to this year's ATHN update, highlighting the exciting activities ATHN has pursued since our last get-together. My name is Michael Recht. I am a pediatric hematologist seeing patients and families at the treatment center at Oregon Health and Science University in Portland, Oregon. I am also ATHN's Chief Science Officer, the person in charge of leading ATHN's research efforts. Joining me today from ATHN are Nikki Hirsch, our Senior Director of Research Programs, as well as Moses Miles, our Chief Operating Officer. At the beginning of any scientific talk, it is standard to list all potential conflicts of interest a speaker may have in order for you, the people in the audience, to judge whether any potential bias by a speaker may prejudice the information the speaker is delivering. As you can see on this slide, neither Nikki nor Moses have any disclosures to declare. However, I have a long list of disclosures as I have been involved with clinical research sponsored by our industry partners for over 25 years. Although it is standard to list potential conflicts of interest that have occurred over the past 3 years, I have taken to list all pharma companies with whom I have worked throughout my entire career to be as transparent as possible. I also list any board of directors on which I am currently serving, as well as my employers. If you have any questions about my disclosures, I would be happy to take any questions at the end of today's sessions or at any time in the future. Here's what we're going to cover today. I'm going to start by talking about the importance of medical research and how it has advanced our field. We will then talk about two of ATHN's larger research projects. Nikki will discuss ATHN Transcends, our new long-term natural history study. I will then give you an update on where things stand on My Life, Our Future, the collaboration between ATHN, NHF, and BloodWorks Northwest in Seattle. Finally, Moses will discuss ATHN's new patient-facing cell phone application, Robust Health. Thank you for attending today's session. I think you're going to enjoy our presentation. So, before we can talk about why I think it is important for individuals to participate in medical research, we have to understand what medical research is. At its most basic, medical research utilizes a systematic approach to asking questions with the goal of establishing facts and creating new knowledge. There are three types of medical research. Basic, or preclinical research, is what you think about when you see people in white coats working in laboratories. This type of research can use chemicals, or cells, or small animals like fish or mice or rats. And any promising results are then moved to what we call translational research, better known as bench to bedside. In this type of research, a new medicine or device may be tested on large animals like dogs or non-human primates. Finally, if things still look good, the research will progress to what we call clinical trials. Clinical trials can be broken down into 4 phases. Phase 1 trials assess safety only. They are not designed to see if a new medication or device works or is beneficial. The reason phase 1 trials exist is to ensure that no new medication or device moves forward in development if it causes harm. Phase 2 trials continue to look at the safety of a new intervention, but add on the question of whether something works or doesn't work. The knowledge gained from phase 1 and 2 studies are combined, and phase 3 studies are conducted. These are the kind of studies you read about in the newspaper when large groups of people receive a new medication or a new combination of previously used medications, or undergo a new procedure, or use a new device. For example, the studies looking at the safety and effectiveness of COVID vaccines were all phase 3 studies. The results of these phase 3 studies are then presented to regulatory bodies across the world to determine if a new intervention should be approved for use. In the United States, the Food and Drug Administration, or FDA, is the organization that makes these determinations. If a new therapy is approved by the FDA, that does not mean research is over. Phase 4 studies look at questions around how to best use new therapies and/or the long-term effects of approved therapies. If an FDA-approved drug is to be studied for use in a different condition than which it was initially approved, a new phase 3 study must be performed. So now that we understand the types of clinical research, let's chat about why I feel it is so important to participate in clinical trials. To make my position as clear as possible, participation in clinical trials is the only way we are ever going to learn about new therapies for a particular condition. Clinical trials are at the heart of all medical advances. Without clinical trials, we would never have new medications, new combinations of existing medications, new surgical procedures, or new medical devices. The benefits for an individual to participate in clinical trials really is to help others. There is the possibility that participating in clinical trials will be beneficial to the participant in the trial, but that can never be guaranteed. What can be guaranteed is every clinical trial is going to move science forward, whether the intervention under study ends up being safe and effective or not. Helping others with similar medical conditions is, to me, the real driver of agreeing to participate in medical research. In addition, if you are someone who likes the latest and greatest, participating in clinical trials allows you access to the newest therapies available. However, that has to be balanced by the fact that we may not know whether these newest therapies actually work, as well as existing therapies, or are safe in the long term. Now, there are people who are just not comfortable with the idea of participating in medical research. And my response to that is absolutely okay. I don't ever want anyone to feel they are being coerced into participating in clinical research. The idea of participating in clinical research should be made after discussion between you and your providers, as well as those whose opinion you trust. The next 2 slides list all of ATHN's current and past research projects. As many of you know, ATHN was founded about 14 years ago as a technology company to help the staff at the United States Hemophilia Treatment Center Network centers collect the data required by our federal partners. What individual ATHN-affiliated clinicians and investigators discovered was ATHN systems were a convenient and consistent way to collect research quality data for the population we serve. Starting at ATHN 1, which looked at heart disease in the hemophilia population and was begun in 2016, we are now through ATHN 16, which is ATHN's first phase 4 interventional study looking at the safety of a new recombinant factor 7A, 7-FACT, when used to treat bleeding events in people with hemophilia A or B complicated by inhibitors. Other ATHN studies have looked at the risk of inhibitor development after switching to new factor products, the risk of radionucleotide synovectomy, the outcomes of those with hepatitis C virus who underwent therapy, figuring out the genetic differences in those individuals affected by rare bleeding disorders, and looking at outcomes of individuals with hemophilia who have underwent liver transplantation because of hepatitis infection. And finally, the use of a new class of oral anticoagulation medicines in children with blood clots. ATHN has also been running natural history studies, which means we are collecting specific data on how people are being treated currently for their hemophilia or von Willebrand disease. The benefits of these natural history studies is that they are long-term, in our case, up to 15 years. By participating in these natural history studies, the participants and ATHN investigators will be able to determine the long-term safety and effectiveness, as well as how people feel about their current therapy. ATHN's newly launched natural history study, ATHN Transcends, will bring together and harmonize the way we collect data. I would like to introduce Nikki Hirsch, ATHN's Senior Director of Research Programs, to describe ATHN Transcends. Nikki?
Hello, I'm Nikki Hirsch. I'm the Senior Director of Research Programs for ATHN. Thank you for joining us today. I want to take the next few minutes to update you on a research design that ATHN has launched called ATHN Transcends. The ATHN team that has helped create and launch this study includes Carrie O'Neill, who is the lead clinical project manager for the study, Dr. Michael Recht, who is the Chief Science Officer at ATHN, and myself as the Senior Director of Research Programs, and many, many more people within ATHN. ATHN Transcends is a natural history cohort study of the safety, effectiveness, and practice of treatment in people with non-neoplastic hematologic disorders. The foundation of this study consists of 7 cohorts, of which eligibility is defined by diagnoses. These cohorts include, from the middle going outward, hemophilia, von Willebrand disease, thrombosis thrombophilia, rare bleeding disorders, congenital platelet disorders, bleeding not otherwise specified, and non-neoplastic hematologic conditions. This last cohort has been included for futuristic thinking for other disorders that would not fit within one of these cohorts, such as sickle cell or ITP. The design of the study is that cohorts can have study arms and modules that are connected to them, so that we can address more specific research questions and collect data to address those questions. Currently, we only have 4 study arms in ATHN Transcends. Under the hemophilia cohort lies the PUPS arm, as well as the hemophilia natural history arm, which was formerly known as ATHN 7, the hemophilia natural history study. And the last arm under the hemophilia cohort is the hemophilia gene therapy outcomes arm. This was formerly known as ATHN 14, the hemophilia gene therapy study. Also under the von Willebrand cohort, we have the severe VWD natural history arm. Now, to date, the only cohort that is open is the hemophilia cohort, and the hemophilia natural history arm is the only arm open under that cohort. It is currently dedicated to transitioning those participants from the ATHN 7 study into this arm of ATHN Transcends. Now, the study is planned to be open for 15 years, so other cohorts and arms are planned to be open throughout those years. We anticipate to enroll at least 3,000 participants over this time. Now, you're probably wondering why we choose this type of research design. Well, let me tell you. The major benefits of this design from a scientific perspective is that it allows for data harmonization across all cohorts, as well as the ability to compare data across cohorts. And the dissemination will be prioritized, allowing for all treatment centers to participate over time. Now, the primary objective of this study is to determine the safety of therapies and treatment for the study population. So safety will be measured by events in the European Safety Surveillance or EUHAS program. These include allergic or other acute events, treatment emergent side effects of therapy, transfusion transmitted infections, inhibitor development, thrombosis, cardiovascular events, malignancies, neurologic events, and death. We will also be collecting adverse events of special interest, which include the occurrence of thrombotic microangiopathies, injection site reactions in cases of potential drug-induced liver injury. We're also collecting the development of anti-drug antibodies to be measured and confirmed if feasible and when testing is available. Thirdly, we're collecting severe unanticipated bleeding and as well as hospitalizations. Now, the secondary objectives of ATHN Transcends is to establish a platform for study arms and modules, which is what we did, Also to describe bleeding event rate, changes in overall bleeding, as well as results on validated measures of bleeding, such as ABR, ISTH bleeding assessment tool, and the pictorial bleeding assessment chart, also known as PBAC, for applicable diagnoses. We're also going to be describing medication dosing regimens, as well as real-world effectiveness of therapies using healthcare utilization, patient-reported outcomes, and patient goals, called GOAL-HEME. The ATHN Transcends study also collects samples for a biorepository for current and future research. Now, let's get into a little bit more information about the ATHN Transcends study. The study itself includes 4 types of visits. So the first visit is the baseline visit. This is where we're going to have signed informed consent, review of medical history, bleeding events, and treatment plans, as well as completion of patient-reported outcomes and laboratory assessments. The visits next will be conducted quarterly. These can be done in person or over the phone to review possible adverse events, medical events, bleeding events, or changes in your treatment plan. Next are annual visits. These are in-person visits to be conducted to obtain the same information as your quarterly visits, as well as completion of patient-reported outcomes and inhibitor testing for applicable cohorts or arms. Now, the final visit is the study exit. That includes the same assessments as done annually. Therefore, it should be done as an in-person visit because there are completion of patient-reported outcomes and inhibitor testing as applicable. Now, throughout the study, there can be ad hoc visits that occur for clinical indication. As mentioned earlier, ATHN Transcends is currently only enrolling those participants enrolled in ATHN 7, the hemophilia natural history study, and it is only open at those centers where those participants are located. However, exciting news is the study will be rolling out through the treatment center network through 2022. So please ask your treatment center about this study if you're interested in participating. Lastly, ATHN has undertaken a renewed effort to ensure results of ATHN studies are communicated widely and is not limited to just medical journals so that patients have more access to this data. ATHN is working closely with NHF to develop a communication plan to ensure the results of these studies and other projects within ATHN are regularly distributed to the bleeding disorder community. Thank you for your time.
Thank you, Nikki, for that informative talk summarizing ATHN Transcends. If anyone has questions about ATHN Transcends, we will all be available during the discussion session at the end of this presentation. Now, I'd like to give you an update on where things stand in the MLOF RR Future project. As many of you are aware, MLOF RR Future is a collaboration between ATHN, NHF, and Bloodworks Northwest. Initially, this project received funding from BioVeritiv. The goal of MLOF RR Future was and is to bring a national program to the hemophilia community to offer free genotyping or figuring out the genetic differences in the factor VIII or factor IX genes in those people with hemophilia and their family members to improve hemophilia care by increasing knowledge of these bleeding conditions and to build a foundation for scientific breakthroughs. Sound familiar? MLOF RR Future meets all the criteria for clinical research I previously discussed. To date, over 11,000 people have participated in the MLOF RR Future project. Enrollment was completed in 2017. Of the over 11,000 people enrolled, just under 8,000 were people known to be affected by hemophilia, and just over 3,500 were women who were known to have hemophilia, were carriers of hemophilia, or were potential carriers of hemophilia. Of the over 3,500 women who agreed to participate in MLOF RR Future, just over 2,500 were confirmed to have variations in the factor VIII or factor IX genes. And of those, almost 2,000 people agreed to be part of the MLOF RR Future Research Repository, sharing their de-identified genetic information to be used for future research purposes. So here are the milestones of the MLOF RR Future project. In 2012, over 9 years ago, enrollment in the project began. What I didn't show on this slide is the fact that this project was in the planning stages for almost 3 years. 5 years after enrollment began, ATHN, again in collaboration with NHF and Bloodworks Northwest, sent out an invitation to researchers to submit proposals for to use both the genetic information in the MLOF RR Future Research Repository connected to all the data that was matched individually from the ATHN dataset, our collection of de-identified clinical facts. In June 2017, a panel of esteemed reviewers from both within the hemophilia research community and those with expertise in this type of genetic research allowed 7 projects to move forward. By the time sample collection was closed at the end of 2017, over 9,000 people allowed their genetic samples to be matched to their clinical data to be used for research. This is the largest resource of this type of data in the world. Of the 7 projects approved in the first round, 2 have been completed and published in the medical literature. Before I dig into the slide, I want to make a brief comment on how to differentiate between genes and proteins in written documents. When genes are being referred to, they're always indicated by being in italics. In this example, the variations in the factor VIII gene being investigated by Dr. Ming Lim at the University of Utah's Treatment Center is indicated by the capitalized and italicized F and 8. If Dr. Lim was assessing the actual factor VIII protein, that would be indicated either by completely spelling out factor VIII with the 8 as a Roman numeral, or by using the abbreviation FVIII. In her project, Dr. Lim looked to see if there were any specific variants of the factor VIII gene associated with inhibitor development in people with mild or moderate hemophilia A, people we don't normally associate with developing inhibitors. She found 77 men and no women with mild or moderate hemophilia A out of a total of 2,557 in the MLOF RR Future Research Repository who developed inhibitors. She found a total of 46 different factor VIII gene variants, 41 more than had ever been previously reported. By identifying people with moderate and mild hemophilia at higher risk for inhibitor development, we may be able to intervene to lower the risk of having inhibitors in the future. Dr. Zubin Sauna from the Center for Biologics Evaluation and Research, which is part of the FDA, looked to see if genetic variations other than those found in the factor VIII gene could help explain the development of inhibitors in people with hemophilia A. Using genetic data from the MLOF RR Future Research Repository, as well as data that was performed in his own laboratory, Dr. Sauna and his team looked at the genetic variations of a specific immune markers known as HLA genes. Proteins created by the HLA genes are responsible for the regulation of the immune system. You may have heard about HLA as they must be matched when people get bone marrow transplants for cancer, for example. Dr. Sauna found that people with specific variations in the HLA-DP gene had a higher rate of inhibitor development than the hemophilia population in general. His team also found people with 2 specific variations in their HLA-DR genes had a lower rate of inhibitor development. This new data can now be used to generate more complex models to predict inhibitor development in hemophilia A patients. From the first round of approvals, 5 projects are still in progress. You can see there are 2 more projects, one from Drs. Harris and Dumont at Bioverativ, and the other from Dr. Kate Pratt, looking at various genetic variations, which might lead to inhibitor development. Dr. Kamir's project is focused on understanding the differences in bleeding patterns with people with hemophilia with the same factor level, and whether there are any immunologic causes for this variation. Dr. Pipe is studying the role of all of our genes and how they can be used to predict the response to various factor products. And finally, Dr. Jill Johnson is looking at the various genetic factors that might help us predict the amount and severity of bleeding in women who carry an abnormal factor VIII or factor IX gene. But wait, that's not all. The MLOF RR Future Research Repository is collaborating with a federal program called TOPMed. What is TOPMed, you ask? Well, it is a program sponsored by the National Institutes of Health National Heart, Lung, and Blood Institute. TOPMed stands for Transomic for precision medicine. The aim of TOPMed is to provide disease treatments tailored to an individual's unique genes and environment. TOPMed contributes to this goal through the integration of whole genome sequencing or figuring out the entire genetic code in each individual who allows their genetic information to be shared. In addition, TOPMed will be gathering data from metabolic profiles, as well as an individual's protein expression and RNA expression. People who are part of the MLOF RR Research Repository had the opportunity to share their blood samples with the TOPMed program in order to obtain this extra level of information. Once results from the TOPMed testing become available, researchers in our field will be able to dig even deeper into the genetic and environmental makeup of people in the MLOF RR Research Repository to ask more complicated questions that can only be answered with this additional data. A second round of requests for proposals for the use of the MLOF RR Research Repository data occurred earlier this year. All researchers wishing to obtain samples and data had to have their proposals to us by January 15th. At the time of this recording, the proposals are currently under review. We have assembled an impressive group of reviewers who have a deep knowledge of hemophilia as well as genetics. The reviewers are assessing whether the proposed projects are answerable with the samples and data we have. We anticipate letting researchers know if their projects have been approved by July 2021. What I want everyone listening to understand is the progress of the MLOF RR program could not have been possible without the participation of the entire hemophilia community. Samples were gathered from over 100 treatment centers across the United States, and the staffs of those participating sites diligently entered data into ATHN systems so we could match up the genetic information with how people responded clinically to their treatment. In addition, people from ATHN, NHF, Bloodworks Northwest, and the founding funders, BioVeritiv, were the brains behind this amazing project. However, the most important collaborators were the amazing people who were willing to donate a blood sample and share their genetic and clinical data so we can answer these important and interesting questions. We are working with the NHF to come up with a plan on how best to communicate the results of these remarkable studies to you. Now, I'd like to pass the slides to Moses Miles, ATHN's Chief Operating Officer. Moses is going to talk to you about ATHN's new patient-facing cell phone application called Robust Health. Moses, take it away.
Hi, my name is Moses Miles. I'm the Chief Operating Officer for ATHN, and today I'm going to talk to you about Robust Health, a new mobile application for Apple and Android phones that's designed to help you manage your care. Robust Health was built to allow patients to have more insight into their care, and if they choose to allow for a closer relationship between themselves and their treatment center. ATHN partnered with 4 hemophilia treatment centers, each of which had a significant number of patients using at least one of the current bleeding and treatment logging applications. Together with feedback from their patients, we designed and built the Robust Health application. It has several new features, such as individualized goal tracking, which are designed to place you, the patient, in greater control of your care. Before I discuss the key features of Robust Health, I want to ensure that you know that at ATHN, we take privacy and security very seriously. That is why the data that you enter into the Robust Health application will not be shared with your treatment center until you authorize it. If you want to use the application without ever sending your data to your treatment center, that is completely acceptable, and we expect that there will be patients who choose this option. When you're using this application, you're in control of your data and no one else, not your treatment center, and certainly not ATHN. Robust Health has several important features. However, in the interest of time, I will focus on these key features. Bleed and treatment logging via the web and via text messaging, individualized goal tracking, questionnaire and survey capture, and reporting. Like most applications for patients with hemophilia, Robust Health contains extensive support for tracking and logging bleeds and treatments. It does this in 2 ways. 1, via the application itself, and 2, via text messaging. We partnered with RipRoad, a company that has staff who create the text messaging infrastructure for AT&T and American Idol to make this possible. The text messaging feature will be available later this year. Earlier this year, we added a goal goal tracking component to the application treatment centers use to manage patient care. This component enables treatment center staff and patients to develop goals that are specific to the patient and to track those goals over time. If you opt to share your data with your treatment center, those goals will be made available through the Robust Health application, allowing you to update and track your progress towards achieving those goals. Often your treatment center will need for you to complete a questionnaire or survey for a study or a surveillance project. If you opt in to share your data with your treatment center can send those surveys directly to you to be entered via the Robust Health application. We feel it is critical that you as a patient have a greater insight into your health and care. The reporting capability of the Robust Health application will give you that insight. As with other features of Robust Health, we need your feedback on how to make the application better, and this is especially true here. We need to know from you what information would give you greater insight into your health. If you have any questions regarding Robust Health or any of the other topics we covered today, please don't hesitate to email us at one of these email addresses and we'll get back to you. Thank you.